Vengat, please give YouTube live.
Initiated, sir. Initiated, sir.
Okay. Chief Guest, sir, please unmute your video on mic. Vengat சாரோட வீடியோ அன்மியூட் பண்ணுங்கப்பா. சீஃப் கெஸ்ட் சாரோட. Sir, can you hear me, sir?
Yeah, I can hear you. Yeah, I can hear you.
Okay, sir. Okay, sir. Sir, you are in office. Am I correct, sir?
Yeah, I am in my office. Yeah.
Okay, sir. Thank you, sir.
Since we are staying inside the campus.
Okay, great, sir. You are staying in a quarters.
Yeah. We can come to the office. Yeah.
Okay, great, sir. Great.
But no students are there. We are alone.
Okay, sir. So we will start within 1 minute, sir. Exactly at 4.
Just like okay, again, Kalyana Kursa Tandovilu, we are daily coming to the office and going back.
It's a correct time to write the proposal and concentrate in chapter other things, sir.
Yeah,
That's right.
But still, okay, that's why I say that, okay. Just like Kalyana Kursa Tandovilu, right? Okay, just coming to the office and going back, okay.
Okay, sir. Yes, sir. Can we start the program, sir?
Yeah, please.
Iswar sir,
yes,
HOD sir?
Vasimala, you can proceed.
Okay, sir.
Yes, proceed.
So, good afternoon to all. Today, our chief guest, Dr. Sri Sivakumar. He's a professor at the Department of Chemical Engineering, Material Science Program, Center for Nanoscience, Indian Institute of Technology, Kanpur. So, thanks a lot for your kind acceptance, sir, during this pandemic period with your busy schedule. So, respected chief guest, respected Dean, School of Physical Chemical Sciences, Dr. S. Kutirani, respected head of the department, Dr. D. Iswaramurthy, and dear participants, I welcome you all for the 14 days event, 14 days FTP program of Material Science and Nanotechnology. So, thank you. Now I hand over the session to Lakshmi.
Good evening to one and all present here. I welcome you all to the 8th day FDP on Material Science and Nanotechnology, MSNT 2020 on behalf of Department of Chemistry, BS Abdur Rahman, Crescent Institute of Science and Technology. There are some instructions to participants. Join 5 minutes before the session to avoid connectivity problems. The session will be live streaming both in Zoom meeting as well as in YouTube. Please mute the mic during the session as the session will not be audible. The participants will be given an opportunity to interact with the speaker by unmuting themselves at the end of the session. Feedback forms will be posted at the end of the session and it will be active for 30 minutes only. And this is for your kind information. As we are getting too many questions in the Zoom as well as in the YouTube, and due to short time, the questions are chosen as per their quality and asked. And so kindly bear with the situation. If any of the participants want to get their answer for the questions, please send a mail to the speaker's mail ID. This is for your kind information. Learning gives creativity. Creativity leads to thinking. Thinking provides knowledge. Knowledge makes you great. With this quote of Dr. APJ Abdul Kalam, we will start our today's session. I cordially welcome the distinguished participants from all over the country to this program. We are delighted to have an eminent speaker, Dr. Sri Sivakumar, Professor, Department of Chemical Engineering, Indian Institute of Technology, Kanpur, Uttar Pradesh. I extend a warm welcome to the speaker. Today he is going to present on the topic Nanoengineering Materials for Biological Applications. Before starting the session, I would like to introduce our speaker. Dr. Sri Sivakumar has obtained his BSc degree in chemistry from Nanoengineering Sundarnar University, Tirunelveli in 1993. BSc Tech from University of Institute of Chemical Technology, Mumbai 1997, and MSc Tech from University of Institute of Chemical Technology, Mumbai in 2001, and PhD from University of Victoria, BC, Canada in 2006. He worked as a postdoctoral associate in Center for Nanoscience and Nanotechnology, the University of Melbourne, Australia from 2006 to 2008. Then he joined in Indian Institute of Technology, Kanpur, Uttar Pradesh as an assistant professor in 2008 and became an associate professor in 2014 and working as a professor since 2018 in the Department of Chemical Engineering. He has published more than 80 papers in international high-impact journals. He has filed 15 patents, published 5 book chapters. He has delivered a number of invited talks at various institutes. He bagged several awards, including a Professor C.N.R. Rao Award for Outstanding Research in 2014, IIT Kanpur, Class of 1979 Fellowship 2014, IIT Kanpur, and Dr. Raj and Neera Singh Chair from IIT Kanpur. He has produced 11 PhD students and many master project students. He has completed several projects from DST, DBT, and other funding agencies. His field of specialization has been focused on the synthesis of nanomaterials for drug delivery, bioimaging applications, and solar hydrogen generation, etc. With this brief introduction, I hand over the session to the speaker.
Thank you so much for the kind introduction. I also thank Dr. Vasimali for the kind introduction and other organizers for the this nice arrangement. So as you aware, everybody aware, this is a very difficult time and somehow we should connect it with each other. This is the one of the nice platform, though, which is we are not seeing it personally, but at least which is we are seeing over virtual platform. In order to save the bandwidth, can I stop my video? Can I stop my video for because sometime okay?
Yeah, yes, sir. You can stop.
Sir,
No
you
problem.
proceed.
You
I
can
will
stop.
stop my video.
Yes.
So in another 45 or 50 minutes, I'll be talking about the nanomaterial, which is we are doing it in our lab for the last 10 years, in particular, especially for the biomedical applications. There. So as you know, okay, there's some problem in the slide changing. Okay, there is some problem in the slide changing, which is I'm stopping the share and then I'll come back again.
No issue, sir. You can.
One second, I will come. Just a minute.
Yeah, no problem. Dear participants, kindly wait for a few minutes due to technical issue. So for the speaker, it is rectifying that issue. Kindly wait for a few minutes.
That now, can you able to see the screen now?
Yes, sir. Yes, sir. Put full screen, sir.
Yeah, I'm going to the full screen mode. Is it changing now?
Yeah, it's changed. Yes.
So,
You can proceed.
sorry for the small problem, technical issue. As you know, the different nanostructures have been applied in the different biological applications. I'm going to talk about two important things. One is the drug delivery and the bioimaging. The another important one is the biosensing. So various nanomaterials have been exploited. All these biomedical applications. So since which is I'm going to talk about the drug delivery and the bioimaging. So I just want to give some importance of the nanomaterial, why nanomaterials are considered as one of the potential material for biomedical application. So this is the sixth grade problem, which is we might have done it in our mathematics course. So there is a 1 meter cube which is present there. So if I want to carve 1 nanometer cube, how many number of cubes can I carve it from there? And what is the surface area, total surface area of that number of cubes? So from this one, we can easily find out that it is something around 10 to the power 27 number of nanocubes can be carved from this 1 meter cube, as well as the total surface area, which is going to be something around 6,000 square kilometers. If I compare that surface area with the 1 meter cube, that's going to be only 6 meters square. So the surface area has enhanced exponentially. So that is approximately equal to 1/7 of our Haryana state. So this 1 meter cube, I can easily keep it on my table. So now the question arises, why the surface area is important, right? So that is because the surface area decides the many physical property, for example, optical property or magnetic property or mechanical property. Because of that huge reduction in the size, which enhances the surface area, that change the property of the material from nanomaterial compared to its bulk counterpart. That is the power of nanomaterial. Because of this, the change in the property, those property can be exploited in the biomedical applications. Now, what is bioimaging? Bioimaging, it involves the imaging of the organelles, tissue, or at the molecule level. So especially in particular, why do we need bioimaging? For example, if we want to study the interaction of the various biomolecules or the signaling pathway or diagnosis of a particular disease. Especially in particular disease I am taking, since I am going to talk about the cancer. So size and location of the tumor we want to find out. What is the cell proliferation rate that has to be found out, and in what deeper the tumor is present, that is the tumor in the deeper tissue, as well as the cancer therapy, that is the tracking of the therapeutics. So all these things which is important for that, which is we need the bioimaging agent. So in that case, nanomaterial can be used in the bioimaging. Since the nanomaterial, since it is going to be used for the bioimaging, especially in the in vivo level bioimaging, so we need to apply this nanomaterials into our body. So first thing, that is the challenge which is comes in the in vivo delivery of the hydrophobic nanoparticle. What I mean by hydrophobic nanoparticle, more than 90% of the nanoparticles are prepared under hydrophobic condition, because we want to attain the highly crystallinity. Because of the highly crystalline nature, their magnetic property or optical property can be highly enhanced when compared to its amorphous nature. So since it is going to be prepared at a high temperature and at the hydrophobic condition, so these particles cannot be used under in vivo condition, because it can get agglomerated. Since because of the agglomeration, it can get cleared by the macrophage phagocytotic system. So now, since cannot be applied, all the hydrophobic nanoparticle can be converted into hydrophilic by using the surface modification reaction. So the one problem is solved, but during the surface modification reaction, it may lose their desired imaging property, especially in particular the fluorescence quenching. It can happen because it is going, water is one of the quenching molecule which can quench the fluorescence. So now, third one, the challenge. Since the nanoparticles are going to be uptaken by the cells, so we have to understand at which mechanism the each nanoparticle is going to be uptaken by the cells. That means that we need to understand the uptake mechanism of each nanoparticle by the different cells. So that is going to be entirely depend upon the surface chemistry, shape, and size. So I want all the participants to note down this uptake mechanism, because I am going to talk about this one at the later stage of my talk. Third one, the next one, the other challenges, the particle should be having the biocompatibility. It has to cross different biological barriers, for example, blood-brain barrier, and it should have an efficient biodistribution. And the very important part is that it should be cleared from the body. And since it is going to be in the blood, should have a prolonged blood circulation with the adequate payload at the desired site. So these are the challenges which is any nanoparticle which is used for bioimaging, or if it is going to be applied for it is going to be used for drug delivery applications. So what we thought is that whether we can circumvent some of the challenges by using it one of the unique approach. The circumvent, what is the challenge? We want to deliver the hydrophobic nanoparticle. We do not want to deliver the hydrophilic. Since it is going to be having a higher optical or magnetic property, we want to deliver the hydrophobic nanoparticle. The second challenge is that whether the uptake mechanism can be a universal uptake mechanism irrespective of what is the nanoparticle has been used. So we do not need to worry about every time, we do not need to worry about the mechanism. And other challenges, biocompatibility and the efficient clearance and prolonged blood circulation, all these things, whether it can be tackled by a single approach. Before going into that one, so I just want to introduce what are the different kind of nanoparticles have been exploited in the bioimage. The one is that quantum dots, that has been well established material, but it has been used in the in vitro level. It cannot be used in the in vivo because of the inherent toxicity. For example, cadmium selenide or lead sulfide or lead selenide. These are all which is going to be, these are all can create a toxicity there. The next one is the gold nanorod, which has been exploited. But there are controversial reports which is available that gold nanorod also can induce the toxicity. The next one is the lanthanide ions. We know that lanthanide ions, which has been the lanthanide, which is present in the bottom of the periodic table, which is lanthanide ion, which can have a very high fluorescence property. So apart from that, there are organic dyes have been exploited in the bioimaging application, but the inherent problem is that the photo bleach. For example, the one which is there in the bottom of the one which is having the green emission, which is coming from Alexa fluoro 488 dye, which has been photo bleached after 180 seconds. So since the fluorescence is photo bleached, we have to do the imaging within a certain period of time. That is not possible. So all these challenges which can be tackled by the lanthanide ion, that if we prepare the lanthanide ion doped nanoparticle, which may useful in the bioimaging application. What is the speciality of lanthanide ion? As I mentioned, that it is present in the bottom of the periodic table, and they all have the unfilled 4f shell electrons. Since it is all unfilled one, so they have a very enhanced photostability because the recycling is happening within the 4f shell. So these electrons do not involve in the any bonding one. Because of that one, they have a enhanced photostability. The second one, it is having a non-overlapping absorption and emission spectrum. What I mean by that non-overlapping absorption emission means you can excite it at UV region and you can get the emission in the near infrared region, or you excite within the visible region and you can get the emission in the near infrared. So you excite with the 300 nanometer, you can get the emission 600, 700, 800, like that you can get it. So their absorption and emission spectrum are there's no overlap between these two. The third one, their lifetimes are range from micro to millisecond. This is a very, very important one for the lanthanide ion because these transitions are forbidden transitions. So this is one important part advantage in the bioimaging, since the lifetimes are going to be very high, so you can easily differentiate the background signal from the signal coming from the lanthanide ion by using the time-gated fluorescence spectroscopy. The fourth one, they have a emission from UV to NIR. That means that especially they have the emission from 300, 400, and 500. All these emissions which is available, and especially the 700 to 1350 nanometer, which is called as a biological window, in which the tissue is transparent. That means an excitation light and emission light can go much deeper. There will not be much scattering will be present. Finally, it is having a low toxicity because those have been already exploited in the biomedical application. For example, gadolinium, which is used in the MRI for a large amount which has been. So though at the very high level, it can create the toxicity, but low level, it will not give you a toxicity. These are all the importance of the lanthanide ions. Apart from that, they have a very unique optical property. The first one is the Stokes shift fluorescence, that is the conventional fluorescence. You excite with the higher energy photon, one higher energy photon, that is a 400 nanometer, the bottom left one there, and you can get one lower energy photon, 540 nanometer. So this is, in this case, the emission which is going to be, the quantum yield which is going to be 100%. The next one is the upconversion fluorescence, where you excite with the low energy photon, but you will get one higher energy photon. For example, the middle one, which is called as, you excite with the 980 nanometer, the electron from the ground state, it can go to the excited state E1. Because of the enhanced lifetime, excited state lifetime, it can take one photon from the laser or from the through energy transfer mechanism, which is, I am not going into that detail, which can go to the next level E2, and then you can get one lower energy photon. Here the theoretical quantum yield can be for 50% there. But this is the one which is I am going to talk about, because Here you are exciting with the near infrared so that your background signal from the biomolecule will not get excited. We will get only signal from the lanthanide ion. The third one is the quantum cutting. The quantum cutting is the one you excite with the one higher energy photon, that means the near UV energy, and you will get a two lower energy photons are there. So out of one photon you get the two. So the quantum yield, theoretical quantum yield can be 200%. So all these things can be exploited in the lanthanide ion. Because of that one, we are interested in lanthanide ion doped nanomaterial for biological, especially bioimaging applications. So now I am coming directly to the problem which is here. Right? So this one, which is the work which is started by Haider, who is who is my PhD student. He finished, and then continued by again. Now he has finished his PhD, and then followed by Dr. Jayashree. She is also my PhD student. So as I mentioned that we want to deliver hydrophobic nanoparticles. So what we did here is that we made a nano capsule. The nano capsule, either it can be carbon or polymer capsule, which are being considered. Within the cavity of the carbon or polymer capsule, we have synthesized this nanoparticle, lanthanide-doped nanoparticle, for example, lanthanum fluoride or gadolinium fluoride, or any particle which is we can load it inside. So the another advantage here is that we can load multiple type of nanoparticles. For example, lanthanide-doped lanthanum fluoride, which can give you the fluorescence emission. Lanthanide-doped gadolinium fluoride, it can give you the magnetic resonance imaging property. So we can do the fluorescence imaging as well as the MRI imaging. So this is called as a multimodal bioimaging. Two different imaging technique can be exploited. So the advantage of both the techniques can be exploited in this case. So what we have done is that we prepared the polymer capsule, loaded multiple type of nanoparticle inside, and did surface modification of the capsule with the polyethylene glycol because of the stealth property, and decorated with the antibody for the targeting one. So this is a kind of a generic approach which is being proposed for the bioimaging application with the universal uptake mechanism. That one I will talk about that one little later. How do we did it? So after doing it, we analyze the in vitro, in vivo toxicity, in vivo bioimaging, and primary preliminary immunology studies. All these things which we have. Now, how do we prepare this capsule? First, I am going to talk about the polymer capsule. The polymer capsule can be prepared by layer assembly. So that is the silica which is having a negatively charge. So we take the negatively charged silica, put it into the positively charged polymer, that's the polyelectrolyte. So because of the electrostatic interaction, positively charged polymer goes and then absorb it onto the negatively charged silica, and then we have a first layer open. Since it is going to be having a residual positive charges, we put it again into the negatively charged polymer. So we will have another layer over that one because of the electrostatic interaction. Now we have one layer positive charge, one layer negative charge. We repeat this process to do the many number of layers. So in this case, typically we did it for 8 layers. Now we have a core shell particle. What I mean here is that polymer wrapped silica particle. So now in order to create the cavity inside the capsule, we remove the silica by etching it with the hydrofluoric acid. Now we get the capsule here. So what is the advantage of this approach? This approach here is that I can play with the polymer chemistry such a way that the polymer can be responsive either to the pH or enzyme, glucose, light, ultrasonics, anything which is there. Because of this stimuli, the capsule can break, and whatever encapsulated inside the polymer capsule can deliver inside the cell. So this is just like a capsule which is having inside the cargo which is going to be present there, and you can play with the polymer chemistry. This is a 25-year-old technique which is being done. So now what we did is we take this polymer capsule, put it into the nanoparticle precursor. For example, gold salt, you take it. So since the salt which is having an ion, This ions, okay, the polymer capsule walls are porous. Porous in the sense it can allow only the ions which is having that. So it will react within the cavity of the polymer capsule and the nanoparticle will be formed inside the polymer capsules. So once it is having the reaction which had happened, when the particle grows, it attains a certain size, it cannot come out. So what do? You just centrifuge the polymer capsule and remove the supernatant where all the bare nanoparticle or uncapsulated nanoparticle will be present. Then you do the further surface treatment for the pegylation and the antibody. So in this way, we can load multiple types of nanoparticle inside the polymer capsule. Once the capsule which has been prepared, so first thing we want to know whether this capsule have been uptaken by the different cells. In this case, we have taken the cancer cell lines, 4 different cell lines, and then which is we have done the confocal laser microscopy. So the green which is coming from the cell, which are all labeled with the organic dyes, the red color which is coming from the polymer capsules, that where the erbium doped nanoparticles are present there, which gives a red emission there. So from the confocal microscope, it is clear that all the nanoparticle loaded polymer capsules are trapped inside or uptaken by the cells. But whether the nanoparticles have been released or whether it is still inside the polymer capsule, we have taken the transmission electron microscopy. So here in this case, in order to have a good contrast, we have taken the gold nanoparticle inside the polymer capsules. So the bright dots which has been present in the sea, so that shows that nanoparticles are still intact with the polymer capsules. So it has not been released. And if you see the figure B, which is we see a vacuole. In the vacuole, the capsules are present. So that clearly suggests that nanoparticle loaded polymer capsules are uptaken, but capsules or the nanoparticles are released inside the cell. So that is what which is we wanted. We do not want the nanoparticle released inside the cell. The third one, once we did it, as I mentioned, we want to know the uptake mechanism. So what I mean by here the uptake mechanism, you take the cell, cells will be having different pathways so that the particle or the foreign material can go inside the cell. So there are many pathways which are being present there. I will come to that one little later. So you block 1-1-1 pathway by putting the inhibitors. So in this case, what we found is that rotorlin 1, which is inhibiting the nanoparticle uptake. That means that it is going via the macropinocytosis. I want all the participants to note this macropinocytosis because I'm going to come back to this one little later. So once this is an approach which is we have done it. So that polymer capsule approach, which is the polymer capsule act as the nano reactor or macro reactor to encapsulate the particle. But what we found the disadvantage of that one, what we found is that we couldn't able to load any hydrophobic nanoparticle as well as the highly crystalline, high temperature nanoparticle, which is we couldn't able to load it because their capsule getting integrated. So Jayashree comes with another alternative approach. She wants to upload upconverting nanoparticle, which needs typically a very high temperature, for example, 500 degrees centigrade for the reaction temperature to prepare the nanoparticle. So what she did it, she started with the silica. The silica which has been coated with the lanthanum vanadate doped with the lanthanide ion, that is coated with the sol gel method. And then once this has been done, core shell particle coated with the lanthanum vanadate, she heated this one with the 500 degrees centigrade so that all the lanthanum vanadate will be very highly crystalline. And then she did the layer by layer assembly, which is I have told earlier. Once this has been done, again she gets the silica, all the nanoparticle which has been on the surface of the silica crumble and then it forms as a nanoparticle. So upconverting nanoparticle has been encapsulated inside the polymer capsule. So once this is done, she has characterized it. The A, which is showing the silica lanthanum vanadate coated silica, contrast clearly shows that it is having the lanthanum vanadate. And the figure C, that is the transmission electron microscope. The nanoparticles are inside the polymer capsule from the contrast which is we found it. and we have done the high resolution transmission electron microscope that shows that nanoparticles are highly crystalline. As I mentioned that in this case, we need very highly crystalline nanoparticle, then only we will get a very high fluorescence because of the upconversion. So once it is done, we have done the fluorescence emission spectroscopy that, so we found excited with the 980 nanometer, that is the near infrared, and we get the emission in the visible region, that is an upconverter. So erbium gives the green and red, chromium gives the red, and the thulium gives in the near infrared region. That is a very, very important one because excitation is also in the near infrared and emission is also in the near infrared. That is very ideal case for the bioimage. So once this is done, again, then we want to know whether these nanoparticles loaded polymer capsules are highly compatible. So we have done the biocompatibility study that is called an MTTSA. All this graph which shows that this nanoparticles loaded polymer capsules are biocompatible. It shows lesser toxicity. So again, we have done the uptake study. All this red green emission which is coming from the nanoparticle excited with the 980 nanometer, that shows nanoparticles are uptaken by the cells. So then now we want to know what is the efficiency of the antibody, whether they, after decorating it with the antibody, whether these capsules are uptaken efficiency has been enhanced or not. For that, which is we have chosen two different antibodies. One is the EGFR, that's an epidermal growth factor receptor. Another one is the HER, that is. So EGFR, that is very specific for H460, but non-specific for HeLa. So the graph at the left-hand side that shows very high specific uptake for the H460, but lesser uptake for the HeLa. Without the antibodies also, the uptake is lesser. That shows the antibodies having a targeted uptake. Similarly, which is we have done it for the HER. HER is very specific for MCF, non-specific for HeLa, and that shows also which is suggesting that the targeting with the antibody can enhance the uptake. So H460 is a 10-fold, MCF7 we find 3-fold increase in the uptake. So now, as I mentioned that whether this capsule approach which can give you the multimodal imaging, we have done the MRI as well as the fluorescence imaging, upconversion imaging. The middle one, which is showing the contrast with the different concentration, that suggests that it is having the capability of the MRI because of the gadolinium ion 3+ ion present, and the green one which is coming present coming with from the erbium ion. So MRI as well as the fluorescence, both the thing it is possible for the multimodal imaging. So now, as I mentioned, I am coming to the uptake mechanism. There are different uptake mechanisms which present in the can be done by the cell. So either it could be a macropinocytosis or caveole mediated or clathrin mediated or clathrin and caveole independent. So in order to use this one, I can block all this pathway one by one by putting certain inhibitors. What I mean is that I shut down the gateway or the pathway of this mechanism there by putting one molecule biomolecule there. So why this is very, very important in the universal uptake mechanism, why which is we are insisting that. As I mentioned, the internalization of particle, it depends upon the size, shape and charge. What I mean here is that if you make a silver or gold nanoparticle, if the size is different, it can go via different uptake mechanism. If the shape is different, it can have a different mechanism. If it is surface modification is different, ligands are different, it can go via different uptake mechanism. So apart from that, uptake mechanism may vary for different cell lines also. So it is very, very important, which is we should have a single uptake mechanism, because each and every nanoparticle we have to understand that that's a very laborious job. So whether our approach which can give you a single uptake mechanism or not, and why, that's the question which is we want to answer. For that, which is we put a specific inhibitor to block different pathway, and then we have done the uptake study. So in the bottom, the right of the one which is having the rotor link, the confocal laser microscope image, that clearly suggests that there is no green emission. That means that nanoparticle loaded polymer capsule are not uptaken. That means that rotor length which is inhibit the macropinocytosis pathway. The other one which is we seeing the green emission. So we quantify it. We found is that these are the one which has been. So that shows the uptake mechanism which is done by macropinocytosis. In this case, we have done with the gadolinium vanadate nanoparticle, lanthanum vanadate nanoparticle. So we change the nanoparticle inside the polymer capsule. It is also goes via the same mechanism. So what does it suggest? It clearly suggests that irrespective of the nanoparticle encapsulated inside the polymer capsule, it goes via a single uptake mechanism. So whether it could be a gold or silver or anything which is inside the uptake mechanism is not going to get changed. This is a very, very important one for the bioimaging or the drug delivery applications. Now we understood the uptake mechanism. We have done the biocompatibility toxicity assay. Now we have done the in vivo bioimaging one. Before do any bioimaging, the first thing is that whether the nanoparticles or how the nanoparticles are biodistributed inside the mice model. So for that one, which is we have taken the tumor induced mice model, and then we have injected a tail vein injection for this one with the EGFR conjugated antibody polymer capsule. What we found, the left hand side graph, it shows a very high uptake in the tumor that clearly suggests that this nanoparticle is having a highly targeted efficiency. Once this is done, we have done the imaging. The right hand side, the bottommost image that shows the again the yellowish emission which is coming, that is from the nanoparticle loaded polymer capsule. So none of the body of the other one which is not showing a much emission. So the emission is coming only from the tumor. So we can easily locate the tumor, and these are all done with the lanthanum vanadate nanoparticles. Now, so all the work which is still now, which is I have talked about the polymer capsule. But I promise I have told already in the earlier slide that we are going to upload the hydrophobic nanoparticle with the polymer capsule. We couldn't be able to do that. Since the reason began is that because polymer capsules get disintegrate in the solvents, hydrophobic solvents, or it becomes swelling. So in order to circumvent that, Amrita and Gargi, they have come with another approach. So what they have done is that they have prepared the carbon capsule. So this carbon capsule, what they have done is that they have taken a solid silica particle, coated with the mesoporous silica, which is having a carbon precursor. So we have done the pyrolysis. All the carbon precursor will convert it into carbon. Remove the silica, and you get the carbon capsule, and then put it into the nanoparticle precursor like earlier under the hydrophobic condition, even up to 300 degrees centigrade. All the nanoparticles, different types of nanoparticles can be loaded. We have taken the transmission electron microscope. So all this one, B, C, D, all the one which is showing different kinds of hydrophobic nanoparticles are loaded inside the polymer capsule. Even with this, we have done one hydrophobic and one hydrophilic. So both the type of nanoparticle also have been uploaded inside the carbon capsule. As said earlier, once it is done, we have done the uptake imaging studies. So all the green and red emission which is coming from the nanoparticle, and done with the different types of cancer cells. So now, once this is done, we want to know the live imaging of the carbon capsule. How this polymer carbon capsules are distributed inside the mice body. That is also which we want to do it in the live imaging. So for that one, which is we have done the SPECT imaging. That is the radio labeled carbon capsule, which is we have done it. We have injected in the tail vein, and then which is we did it. The figure A or the figure D and the figure F, that is from the middle one and then the right hand side one. That so what we found is that the if the particle is bigger, 350 nanometer capsule, most of them it is either it is accumulated in the liver or in the lungs. But there are some which is coming in the coming into the kidney organ. But if the size of the capsule is lesser, what we found is that it is more and more it is going to the heart as well as it is coming to the bladder. That means that the carbon capsule started excreting from the mice body. This is a very good sign because nanoparticle will not get accumulated in that. So which is we have done it. What this is, since this study is done within the hour. So we have done the studies up to 8 days or 7 days, which is we have done it. All the capsules have been excreted from the body. So these are all the studies which is we have done it with the for the bioimaging studies with the Lanthanide-Doped Nanoparticles. Now I am moving to the another part, that's the drug delivery. So as we know, the cancer for the chemotherapy is given. The chemotherapy, it is because of the chemotherapy, it not only target the cancer cells, it target the healthy cells. Because of that one, people get very enhanced side effects. So any cell, which is we know that nuclei is the master regulator of the oncogenic genes which are being presented. So for the anti-cancer, the drug target is lot of drugs which is having the nucleus as the target. For example, doxorubicin, it intercalates with the DNA which is present inside the cells. But if the drug, if it is needs to reach the nucleus, it has to cross many barriers. For example, it has to cross the cell membrane, endosomal landscape, and double layered nuclear membrane. These are all very difficult to cross this many barriers. So the drug efficacy, which is going to be in the order of 10 to 15%. So remaining 85%, that is not in use at all. So what we thought that whether we can design a nanoparticle for to deliver the drug directly to the nucleus. For that one, if we design the nanoparticle, it can increase the therapeutic efficiency. So what are the existing modes which is available in the literature? The first one, there are people used to do it that they decorate the nanoparticle with the nuclear localization signal peptide molecule. If it is going to be there, it will directly target the nucleate. Or you can use a viral vector based delivery, or you can use some kind of an molecular based based approach also, which is people have done it, and some people have done some surface modification. All this technique which is going to be having a challenge because the surface conjugation with the nuclear locally NLS peptide, it may lead some immunological complexity, especially in the in vivo condition. Surface modification is going to be very labor intensive and it may increase the cost. And as I mentioned that different cells may show different response because of the peptide which has been present. Viral vector is a very well known, it can have a pathogenicity or oncogenicity. All this immune it can create the immune response. Finally, the size of the nanoparticle should be in the order of the nuclear pore size. So there is a okay. This is I'm going to talk about a oxymoron one concept here. So now which is I'm putting a cell. In the cell, in the tissue, the bottom part which is going to be having the tumor, where which is you see that okay, there's no the blood cells are not having a tight junction. That's a porosity which are being present. In the top part, where which is you are seeing the green one, the healthy tissue, the red one which is going the endothelial cell are tightly. It is having a very tight junction. There is no porosity. But in contrast, in the tumor part, which is having a porosity. So what does it mean here is that the bigger nanoparticle can go very easily in the tumor part rather than to the healthy tissue. But this mechanism is called an enhanced permeation retention effect. That means that the particles in the order of 200 to 700 nanometer, it has having an enhanced permeation, and then it can also stay inside the tumor part, which is desirable for the drug delivery as well as the imaging application. But there is one disadvantage. As I mentioned in the previous case, if the size is bigger, it cannot reach the nucleus. So there is a trade-off which is you need. So the size should be bigger, but at the same time it should reach the nucleus. So what we thought is that whether if we can make a very soft, bigger nanoparticle, if it is going to be soft, then what it can easily it goes inside this nuclear in the tumor part because of the APR mechanism, and it can go to the nucleus because of the soft nature, it can squeeze inside the nuclear pores. So for that one, which is we have chosen the polymer PDMS. PDMS is the dimethyl siloxane, very well known polymer, very soft polymer, and it's optically transparent, inert, non-toxic, which has been used in many applications. Their Young's modulus is 0.1 GPE, very soft polymer. So it has been used in many applications, especially in the lung mimic or it's implants and everything. It has been used. But the challenge here is that to prepare as the nanoparticle, because the PDMS polymer, it tends to form as the film, but it will not form as the nanoparticle because their surface energy is like that. So what we thought is that, so what is the technique which is we should use? Gargi has come with a nice idea that if we confine this polymer into the pores of a material, and if you do the cross-linking, this nanopart, this PDMS will not form as the film. So what she has done is that, like earlier, she made the solid core mesoporous silica particle and loaded the PDMS. She did the curing and cross-link and everything. She gets the silica. She got the nice 200 or 250 monodispersed, very soft PDMS nanoparticle. With that nanoparticle, we have loaded the drug and then we treated with the cells. So the transmission electron microscope and the scanning electron microscope shows that clearly suggests these particles are highly porous as well as very soft nature. The soft nature, which is I am saying it based on the size, because of the electron beam, right? Okay, it changes its shape. So after done it, we have loaded the nanoparticle with the doxorubicin. So we have done the cytotoxicity assay with the bare nanoparticle, no toxicity, but after loading the drug, which is showing a very high killing efficiency, especially the IC50 value for the HeLa cell, which is much higher. The free dox, which is going to be 1.2, the IC50 value, for the nanoparticle loaded one, that is something around 0.2 there. And similarly, which is we have done it for different cell lines. So after loading the drug in the PDMS, it is showing enhanced killing efficiency when compared to the free drug. We have done the uptake studies, whether the drug reaches the cancer or not. So the left hand side, the nuclei, those are all labeled with the DAPI. And the second middle column, that is the doxorubicin, which is the fluorescent, which gives the inherent fluorescence. And the third one is the merged image. So you can easily clearly see that the more and more the magenta color which is coming, that is coming from the nucleus, that means that the drug nicely reaches the nucleus and it is surrounded by the nanoparticle. Lot of nanoparticles surrounding the nucleus, the yellow color image. So from this one, we can clearly suggest that the nanoparticle reaches the nucleus, as well as we have done the comet assay, that is the tail DNA analysis, which is we have done it. That supports the statement of the MTT assay, which is we have shown, that I show the IC50 value. Here also, which is by found more than 70% increase in the. Once which is we have done, we want to know the biodistribution. For that one, which is we have done it with the non-loaded, that means a very virgin PDMS nanoparticle. What we found it is that in the blood, lot of nanoparticle which has been present. So, and which is we also found in the urine as well as the fecal. So that shows the excretion. So this is done for the 48 hours study. So once all this in vivo study which has been done, now we want to know the tumor regression study. So we have taken the tumor model mice, injected the nanoparticle loaded PDMS. What we found, the bottom part, which is we see a clear-cut reduction in the tumor size. But free drug, there's no much reduction. And now only nanoparticle, no, and only the PDMS, these are all the control samples. But with the nanoparticle, which is we see a reduction. So the magenta one, you see there is no much increase in the tumor volume after 15 or 17 days. That clearly suggests the PDMS loaded drug loaded PDMS nanoparticle, it is having a very high efficiency in the tumor. So all this one, when the Gargi has done it, there are some, when we approach the medical practitioner, So they have a certain issue by using it, by etching it with the hydrofluoric acid for the removal of silica. And that method is not a scalable method. So again, who is the Tata Research Development employee? He has done MTech, now he has finished the PhD. So since he is coming from the industry, so he is interested in the scale up of the nanoparticle. So what which is he has done is that he comes with another method called the nano emulsion method. So nano emulsion method is called then you prepare two nano emulsion. One nano emulsion we have the PDMS base, another nano emulsion which is having the cross link. We mix it together and then cross linking it is happening, and then which is he has done the formation of nanoparticle. But when he done the precipitation and washing it with the ethanol, there's no nanoparticle is forming. All of them are formed as a film. So he has okay. Again, we are very much worried why this is not working, because the confinement is happening and everything, and why it is not happening. So since he is having some molecular dynamics studies background, what he has done is that he has done some molecular dynamics studies. Why this PDMS is not stable? Why it is forming as the film? So when he done it, so what he has done is that he has taken the 0%. This is the leftmost corner one, the 0%. Here only PDMS is present. There is no hydroxyl group present. But when he increase the hydroxyl group 20%, 60%, 80%, so all this PDMS, it is converting into a spherical, or okay, it is coming to some kind of a finite shape which is coming. So what we thought is that okay, if we have a hydroxyl group present onto the PDMS, it will not form as the particle, it will form as the thin film which is there. So what we have done is that same technique. After the nanoparticle is forming it, which is that he has treated with the sodium hydroxide addition. That will hydrolyze it silica, which has been present, the SiO, which introduces a lot of OH group which has been present. After that, so nanoparticles are stable, and then we found the contact angle. Because of the hydroxyl group, the contact angle reduced from 114 to 95. This is the PDMS. So that's a very, very encouraging result. We have done the MTT assay for the toxicity. Once the drug is loaded, the toxicity which has been increasing, but and we have done the uptake study. Here the nanoparticle did not reach the nucleus, but it goes to the mitochondria. So since it goes to the mitochondria, the drug killing efficiency is not that much high when compared to the approach which is we have done it with the silica template approach. So he has done with the different studies to understand confocal microscope with the different cells, but he is not very, very happy. So what which is he has done it is that I'll go to the one. So he has changed the method. Whether if he change the method, whether the nanoparticle can target the nucleus or not. So he has done with the nano precipitation method. So nano precipitation method he has done with the one base, another solution which is the cross-linked, mixing it together. Because of this one, so both the things mixing it together, and then he add the non-solvent with the surfactant. Here the trick is done by the surfactant. Because of the surfactant which is present there, this PDMS nanoparticle PDMS forms as the nanoparticle decorated with the surfactant. So again, he has done all this treatment and everything. He got a very nice nanoparticle which has been present there. In this case, surfactant we use the sodium dodecyl sulfate. Now the nanoparticles are highly monodispersed. The size is around lesser than 50 nanometers. Now he has done the MTTs assay. When compared to the previous nano emulsion method, what we found, the IC50 value drastically it has been reduced. Okay? And similarly for this is for HeLa cell, the left hand side one, the right hand side one for the MCS7. So that's a huge reduction in the IC50 value when we loaded the dogs with the nanoparticle. He has done the uptake study with which cellular compartment nanoparticle goes. He found again it is going to the mitochondria as well as it goes to the nucleus. So it targets the two cellular organ. Mitochondria is the powerhouse of the cell. Nucleus is the regulator of the oncogenic gene. Because of the dual targeting, we found enhanced killing efficiency when compared to the previous one. So we have done again the confocal laser microscope, HeLa cells with the DOX PDMS and the free DOX. We found the DOX loaded PDMS, the top panel is showing the mitochondria as well as the nucleus, but only the free DOX, it goes to the nucleus. Similarly, he has done it for MCF7. We found the similar result goes to mitochondria as well as the nucleus. So once this is done, because of this lockdown regulations and everything, which is we are having some issues in the using the animal. What he has done is that he has created the in vitro tumor model, and then he has treated that in vitro tumor model with the nanoparticle as well as the free DOX. The bottom panel, the LOX-loaded nanoparticle, the left-hand side is the one with the before treatment. The right-hand side, the top, the bottom right is the one is the treated one. So we found the tumor which has been disintegrated, and then lot of cells which are being killed. So with this one, what we come to the conclusion is that so the DOX-loaded PDMS nanoparticle prepared by the nanoprecipitation method has the enhanced killing efficiency when compared to the previous method. So now I summarize. We prepared the nanocapsule, and this nanocapsule acts as a universal nanoparticle carrier as well as the bioimaging agent. So the bigger size soft PDMS nanoparticles, they target the nucleus, but the smaller size PDMS nanoparticles target nucleus as well as the mitochondria prepared with the by the nanoprecipitation method. And we also exploited this PDMS nanoparticle for the tissue engineering application, but due to the time, which is I'm not going into that one in detail. Now, which is I would like to acknowledge my students who are all the pillar for all this work, and our collaborator Professor Ashutosh Sharma and Sanjay Gambhir and Andrew and Ashok, and finally funding from DST and DBT, DST UK and Indo US 4. Thank you so much.
Thanks a lot for your excellent presentation, Professor. Very nice presentation, and you have discussed many things, including the fundamentals to advanced level. So we have received several questions in a chatting box from Zoom link as well as a YouTube link.
Yeah, thank you.
Now I request the participants, those who are in a Zoom link, you only have the opportunity to talk with our chief guest. So kindly unmute your mic and talk with our chief guest. Vengat, please check anybody is raising hand.
Hello. Hello. Hello.
Yes, yes, please go on. Yes.
Okay, sir. One thing I want to know that you have prepared PDMS, and how do you measure the contact angle of PDMS nanoparticle?
Sir, chief guest, sir, you just unmute your mic.
Yeah, my mic is unmuted.
Yeah, yes, sir. Yes, please. You can talk.
Yeah. So what we have done is we have taken this nanoparticle and put it onto the glass slide, dry it, and then we have taken the contact angle.
Okay. So it is a good method for measuring contact angle.
You can do that. Yeah.
Okay. Thank you.
Or you can spin coat it also. You can also spin coat this PDMS nanoparticle and then do the contact angle measurement.
Hello, good evening, sir.
Yeah, good evening.
So I am Dr. Pongodi from NGM College, Pollachi. I would like to thank your very nice and informative presentation. But I want to ask one question. Yeah. What's the real-time application of these nanoparticles?
Okay. See, yeah, especially in the terms of the biomedical application, it has to cross many barriers. It has to cross many tests. Phase 1 trial, phase 2 trial, and phase 3 trial, etc. Okay, right? So in our case, we have gone up to the animal studies, and now which is we are planning for to coordinate with the doctor from Sanjay Gambhir from SGPG in Lucknow, but we need to get a lot of clearance to go for this studies. At this moment, whatever which is we have done it for last 10 years, we have gone up to the animal one. It has to go further. And one thing is that we have done some immunological studies, okay, with the Andrew Jackson from University of Nottingham. That also shows a very high immunocompatibility. So with this one, which is we are thinking, okay, to go for the one. So it is not an easy thing to come to the application in the for the human being. It will take a lot of time. Any nanoparticle, it will take something around 6 to 7 years or more than 10 years, okay, to reach that level, commercial level.
Okay, sir. Thank you. Thank you very much, sir.
Any other participants? If you want to talk with chief guest, you can. Okay, sir, I can go for the charting box question.
Yeah.
Dr. Shashank Sharma, sir asked about why lanthanide materials are very important in the field of nanotechnology.
Because of their optical property, because since it's are all the 4F group elements, that 4F shell is unfilled or partially filled that. So because of that partial filament, this electrons, okay, when it is getting excited, okay, they get recycled within the 4F shell. It is not, the 4F shells are not involved in the bonding, okay, right? So because of that, they have a very superior property, optical imaging, okay, especially the fluorescence one. So as such, the lanthanide ion, okay, right, it can be done. We can use it as the bulk material also. For example, lot of okay, the phosphor material, which has been as a bulk material, it is not as a nano material at all. Here the nano material, because of the processability, that's all, nothing. So lanthanide ion, it does not matter whether it is used as a nanomaterial or whether it is used as a bulk material. The property which is going to be almost same. There will be some difference. It is going to be present there. I do not deny that. But only the processability comes from the because of the nanoparticle. That's all.
Okay, thank you, sir. And this question was asked by our MSc student Venkat Ramanan. Sir, you just mentioned the synthesis of different size of particles. Is there any parameter to keep this much size of the particles to inject for the drug delivery application? For example, big size of particles, sometimes it will not able to come through the fecal matters or sweating pores. So can you comment on it?
Okay, so the size is very important, but it's not only determined by the size. It's determined by the shape, determined by the surface functional group which is going to be present, determined by the softness of the material which has been present there. Many factors. But since he has asked the size, I would suggest is that if anything with the intravenous or anything which is that anything size lesser than 100 nanometer is more ideal for the intravenous one. But there are some people who has done is the direct intratumoral injection. There, okay, you do not need to worry about the size. Depends upon the mode of administration. For example, oral administration, you do not need to worry about the size much.
Okay, sir. Thank you, sir. Then this question was asked by Pallavi Pandavan. So I congratulate this madam. Every day, madam asking several questions. I don't know, madam is in Zoom. So if madam is in Zoom, please unmute your mic and ask directly to our chief guest, madam. Vengat, please check. Madam is raising hand. Pallavi Pandavan. Okay, if not, I can go for her question.
Yeah,
Sir,
yeah.
We have prepared nanoparticles in the powder form by using green method. Is it possible to use these nanoparticles for biomedical application?
Okay, this is a very difficult question to answer. It depends upon what kind of a nanoparticle or what is the method which is. So the simple way is that they can do a FDP assay, cytotoxicity assay with the cells. So that will tell you whether it is a toxicity or not. Based on that study, they can take it to the further.
Okay, sir. And another one question, this one was asked by my PhD research scholar, Immaculate Mercy. What kind of drug delivery is available for coronavirus disease? And comment on what kind of research are going to find the coronavirus vaccine.
If the drug is available, I would have directly come to Crescent College to give the talk. I'm very happy. Okay, you are going to come. To my knowledge, okay, there is no drug or anything which is available. It's very well known to everybody that. But as you know, how much which is you know that much which is I also know it. Okay, the vaccines and everything is all in the development that. Once we get the result, then only which is I can comment on it. Okay, there are lot of literature which is coming. Okay, they have tested this one, they have tested this one, but unless and until if anything which is commercialized, then only which is we can believe.
Yes, we understood, sir. And this question was asked by Professor Kimshuk Desai. Plant extracted mediated nanoparticles are useful one for drug delivery application. Is it useful material for drug delivery application? Synthesis of nanoparticles from plant.
It has to be tested. They have to load the drug and then test it. Then only which is it can be commented. But it can be tried.
Okay. So now again I am giving opportunity for the Zoom link participants. Please unmute your mic and talk with our chief guest. We have received several questions from chatting box, but I can give the priority for those who are in Zoom link. Abhilash Vengat, please check. Yes, please, sir.
This is Venkat from IIT Kolkata. Shiva, very nice talk, very nice presentation.
Venkat, how are you?
Hello, sir. How are you, sir?
Thank you so much
I did not expect that you are there. Okay.
Me too, sir.
I'm there from the beginning. So of course I know your work and things like that. Very excellent talk. Very nice. Thank you so much. Hi Vasumalai. Thanks for arranging.
Hello, sir. How are you, sir? Yes, sir.
I'm
it's surprising for me. You are also a participant here.
Oh, I'm here. I attended today's talk, of course, Shiva's talk. There
yes. Thanks a lot for coming, sir. With your busy schedule, you just came and listen to our talk.
Pleasure.
And through this online platform, I would like to say thanks for our speaker. I found the speaker through our Venkatramanan sir only. He is my first friend. And Siva sir is the friend of Venkatramanan. So through that network only, I found him. So again, I thank Venkatramanan sir to identify this.
Okay. So when you say something, I cannot deny it at all.
Yes.
it's a pleasure. Okay. So, Shiva, and I know that. So everybody gets. I know Shiva works on something very important and interesting thing. So I am very happy that it reaches to our people, no, from nook and corner of our India.
Yes, sir. Very excellent talk and
that way I'm really happy.
Yes. Yeah. So if you want to ask anything, please.
No, I don't have any questions. I just want
to
say.
Okay. Thank you, sir. Thank you.
Carry on.
Thanks a lot, sir. Thanks a lot for your wishes and fullest support, sir. Thank you, sir.
Thank you. Thanks, sir.
Chief guest, sir, again, I can go for the one more question. It is from Abhilash, our second MSc chemistry students. Sir, I have read one article, copper nanoparticles coated nose mask is the prevent and kill the coronavirus. Please explain the mechanism how copper nanoparticles are killing this type of virus.
Oh, okay. So I do not know because this is a very new one but there are some reports which is available for copper and silver. Okay. So they create, they form this reactive oxygen species, ROS and everything which has been generated. So maybe that may be the mechanism, I do not know.
Okay, sir. Thank you, sir. And Subaraja, sir, asked about which size of nanoparticles across the blood-brain barrier?
Oh, blood-brain barrier, okay, anything in the order of 10 nanometer.
10 nanometer. Okay, okay, sir. And another question, Sasang Sarma, what is the bulk nanomaterial synthesis process for drug delivery?
Okay, there are many methods which is available there. Okay, even just by precipitation, many methods are there. Colloidal synthesis, many are there. Okay, right?
Okay, okay, sir.
Many are there.
Okay. Another question, Sakin Kumar, how bigger size of nanoparticle injected into the body, which will affect healthy cell, but you said that smaller particle size are needed for EPR mechanism. Can you explain?
Okay, I said bigger size is needed for the EPR, 200 to 700 nanometer that, okay, right? Because that will not go to the healthy cells, because the healthy cells part, that side which is going to be having a very tight junction, but in the tumor part which is going to be having the leaky part. Okay? So 200 to 700, that is an ideal for the EPR mechanism.
Okay, okay, sir. And one more question from my students, Gokuliswaran. He asked about, sir, different kind of bioimaging is available. For example, green fluorescent, red fluorescent, and blue fluorescence, etc. But these days, for biomedical applications, red fluorescence is novel today. So what kind of nanoparticles is useful to prepare the red emissive nature?
See, there are many particles which can give you the red emission. For example, in the lanthanide-doped one, europium is the one it can give you the red emission. Erbium is the one it can give you the red emission, or holmium it can give you the red emission. Similarly, there are cadmium selenide or different nanoparticles. That also there are some phosphors also. Manganese doped one, because Mn2 plus emission is tunable. It can give you the red emission. So many are.
Okay, sir. Thank you. Then this question asked by Seen Kumar N, Assistant Professor, Jan Xavier Catholic College of Engineering, Nagarkoil. Sir, nano emulsion generally are made by two mixers. Is it suitable materials for thermodynamic applications? Or
Thermodynamic application. What
thermodynamic quantities?
I'm not able to understand
Yeah,
the
me too
question.
also. I
Yeah.
guess it is, he asked about the
It's asking that
photodynamic.
whether the emulsion is thermodynamically stable or not. If that is the question, the emulsion which is we have prepared, those are all thermodynamically stable.
Okay, okay. Thank you, sir. Now again, I am giving the opportunity to the participants. Please unmute your mic and directly talk with our chief guest. Many people are here, so you can utilize this opportunity. Please ask.
Sir, good evening, sir.
Good evening.
Sir, for HeLa cell lines or MTT assay, there is a reference, no, sir. For example, cisplatin is used for anti-cancer drugs. What is the reference for this nanomaterials?
Different cells will be having a different mechanism. Okay.
Okay.
So the particular drug can be active in one cell, but does not necessarily it will be active in another cell, because the drug active mechanism is the killing mechanism can be different.
Okay.
So it can be different for different cells. So every cells will be having a different IC50 value.
Okay. Okay.
The same case is true for nanoparticles.
Okay. Cisplatin also can be used even for this.
Yes, cisplatin is a very well-known drug for the cancer therapy.
Okay. Sir, please tell where are you from? Kindly tell your name.
Sir, I am Dr. Edayanand from Government City College, Hyderabad, sir.
Okay, sir. Great.
Assistant Professor of Chemistry. And another thing is that the lanthanides are generally regarded as radioactive, and therefore they are automatically carcinogenic. How can they be used?
No, it is not. Okay, depends upon the your atomic weight. Whether it is radioactive or not. The one which we are talking about, those are all not radioactive.
Okay, sir. Okay. Thank you, sir. Thank you.
Yeah.
Any other participants? Okay, sir, I can go for the chatting box question.
Yeah.
Laxman Sastri, that sir asked about what is the use of PDMS, polymethyl siloxane?
Yeah. So PDMS it is used in many applications. For example, it is used as an implants in the body. It is used as a as a different organelles preparations and everything, artificial organelles, and it has been used in the coating. It has been used in the shampoo. So because since it's a soft nature and shiny nature, it is used in many applications. The PDMS.
Okay, sir.
Starting
Thank
from
you, sir.
cosmetics to again biomedical application, it has been used.
Okay, sir. Thank
And
you.
it is used in the preparing the microfluidic channels. That is one of the preferred material for in the microfluidic channel.
Okay, sir. And this is my question, sir. Sir, you have performed in vivo study as well as in vitro study. Do you have that medical facilities? Generally, this in vivo, in vitro study, people are need to have a collaboration with medical university or some places.
So we have dedicated cell culture facility, and we have our lab has a dedicated animal room with the different cages, and we have several animals but we are using only the mice models. We are checking it with, and we are in the process of learning to bring the cancer tumor models also, which is we are working on it right now.
Okay, sir. And one more question: you just use the HeLa cells for a cancer cell line, or any other cells you have?
So we have done many HeLa, MCF7, CALU, MM4, there are many which is we have done. We have 7 to 8 cancer cell lines in our lab.
Cancer cells. What about normal cell lines, sir?
Oh yeah, L9 to 9 fibroblast, So we have that. The dendritic cells, macrophages, we have it. Even we can isolate it from the blood. That is also which is
yes, sir. Okay, sir. And this question was asked by Dr. Kotish Chaudhary. Sir, I have worked on several carbon nano quantum dots. During your lecture, you have mentioned that quantum dots are toxic in vivo. How it will be the case of carbon nanodots, sir?
Okay, there are many reports carbon quantum dots which is used in the which is having a very high fluorescence. So it depends upon what kind of a precursor they are using it, and then what is the preparation method. But carbon quantum dots are considered as the biocompatible.
Biocompatible.
In general, but depends upon the preparation technique.
The same one we can apply for bioimaging applications because it is fluorescent.
Carbon quantum dots can be used for bioimaging.
But majority with the carbon quantum dot have been reported in the visible regions as well as UV regions. Is it not a problem?
There are quantum dots which gives the emission in the visible one. They have a tunable emission.
Tunable emission. Okay.
Yeah, they have a tunable emission.
Okay, sir. Thank you, sir. The participants, again, you are getting a chance. Please unmute your mic and talk with our chief guest. We have a sufficient time. Please utilize it. Yes, sir. Yes, please.
Yes, I have not any query for the subject. What I would like to suggest is that there is some.
Sir, your voice is getting break.
Yeah, there is some.
Yes, please.
What is called Pandit Madan Mohan Malviya scheme, under which if you conduct such program, it will be recognized as FDP, and that can be used for cash purpose.
Yes, sure. Thanks for your information. Already we know about that program, sir. We have a plans after unlock. We will apply for funding and we will conduct this type of program. But during these pandemic periods, we thought that this is sufficient one. So we also have that plan to apply for that funding, sir. Sure, sir. Thanks a lot for your information. Where are you from, sir?
I am from Mumbai. From
Please tell your name, sir. Your good name, sir.
My name is Sharat Tajne.
Okay, sir. Thanks for your comment, sir.
Okay.
So participants, anybody, if you want to talk, please do it. Venkat, please see one professor is raising hand, Sharat Dajani. Please unmute, sir, mic. Sir, you can unmute by yourself. We have given the provision. Please. Okay, if not, I can go. Yes, please.
He only talked down, sir.
Oh, okay, thank you. And another question, sir. This is this question from Dr. P.K. Kalita, Professor Rajiv Gandhi University. How can we make a cadmium chalcogenide as biocompatible? Cadmium chalcogenide
Okay,
as
cadmium, we are talking
biocompatible.
about cadmium sulfide, cadmium selenide, cadmium telluride.
Yes,
right?
yes.
As soon as you have a cadmium, I would not, and certainly, okay, I will not take it inside my body, okay. Though, if it is reported that it's a heavy metal. Because if it is accumulated inside the body, it is going to create the problem.
But people are coating the biocompatible material
Yeah,
like
they are
universal.
okay, silica coating, polymer coating, and all these things.
Polyethylene
I would not go for it. Okay, I would not. Certainly, I will
glycol
not.
also they are coating.
Yeah.
Okay. Okay, sir. And this is
Possibly, I am unable to hear your voice.
This is on a general question. It's from my side. Is how many mice you have to take it, and how you people are taking 12 set of mice or 6 set of mice, and they are adding cancer cells for one mice. Am I correct, sir?
You have to create. You have to calculate the power number that is which is we follow that. You should know how many parameters which is you are going to do that, and for every parameter which is you want to check it, you have to choose at least the 5 mice.
Okay.
And you have to have as many as positive control and as many as negative control.
Sir, your voice is getting break, sir. Can you explain one more time,
now,
sir? Yes,
okay.
sir.
What I'm saying that you have to create the power number. You have to calculate. That depends on the number of mice, number of parameters, and the number of controls which is you want to use it.
Okay,
but
sir.
Every experiment you should have 5 mice.
Okay, sir. And for in vitro drug loading and drug releasing, so which instruments you are using, sir, and what kind of
no instrument or anything which is there. We just take it, put it into the buffer, and put it to the stirrer, and then we do it. Or if we want to do it in 37 degrees centigrade, we have a 37 degrees centigrade incubator. You just put it inside. You do not need
and how do you calculate this much drugs are loaded?
Okay,
this much drugs are releasing, sir,
for
from
the
the
Doppler
nanoparticle.
division, we use the fluorescence spectroscopy to measure the drug release as well as the drug loading.
Drug loading. Okay. What kind of receptor you are using, sir?
Receptor means what?
To reach the cells, to reach the cancer cells. So
no,
folate receptor.
Okay. He is talking about folate receptors.
Yes.
We have not used the folate receptor, which is we have used that HER as well as the other antibody which is we have used it.
Okay, sir. Because I am also working in that area, that's why I know that this much level.
Folate one. I'm not very comfortable with using the folate because folate receptor sometimes it is expressed in other cells also. Okay. But still, people use it. Okay, but in this case, we have used the EGFR as well as the HER because these are all monoclonal antibody rather than that one. That's the reason.
Okay, sir. Thank you so much, sir. Participants, if you have any other question, please unmute your mic and ask with our chief guest.
Hello, sir. Hello.
Yes, please, sir.
Yes, sir. Please. Hello. Thank you, Dr. Vasamali, sir. Thank you very much for your wonderful opportunity and wonderful session today. Thank you, sir, Professor Sri Sivakumar, sir, for your excellent and wonderful lively presentation. I have no question, but I have one clarification. See, in order to prepare the lanthanum-based the nanoparticle or the composite component, in order to make a drug delivery and cure the cancer cells. You are using that polydimethoxysilane. Polydimethoxysilane, you just encapsulating this in the nanoparticle. My question is, what is the role for this polymer, sir? What way it interact with the drug delivery system or enhancing, attracting the cancer cell? I want to know such things, sir. Thank you, sir.
I want to clarify here one point here is that the
Yes, sir.
lanthanide-doped nanoparticle, we have not used the PDMS at all.
Yeah.
The lanthanide-doped nanoparticle that PDMS that has been the work which is we have done it. PDMS does not have
Yes,
any
sir. Chief Guest, sir,
relationship
you
with
can
the
talk.
lanthanide-doped nanoparticle. Those two are entirely a different work.
Okay. Okay.
PDMS we have used it because of their soft nature.
Yeah, that's fine. Oh.
Yeah, because of its very very lesser younger modulus.
Yeah.
Young modulus. So which is we thought okay it can reach the nucleus because of the it can squeeze inside the nuclear pores. Yes, it is lesser than 0.1 GPa. Okay, Young's modulus.
Yes, sir. Yes, sir. Yes, sir. Thank you, sir. Thank you very much for your today's wonderful presentation, sir. Thank you, sir.
Thank you, sir. Any other participants? Please unmute your mic.
Sir, Sivakumar, sir. Good evening, sir.
Talk with the chief guest.
Sivakumar, sir. Good evening, sir.
Good evening. Good evening.
Hello.
Yeah, I can hear you, sir. Please tell me.
Sir, is there any general for nano
Yes, sir, you can
this
talk.
one, sir?
Your voice.
Magazine or journals?
No, your voice is not very clear. You just talk.
Sir, general, sir. I am asking general, sir.
Journals, okay. There are many journals on American Chemical Society itself. It is having a ACS Nano, okay, and ACS Applied Nanomaterials, ACS Applied Materials Interfaces. Even Langor it publishes, Chemistry of Material publishes, and Nanoscale which is available, Nano Medicine which is there are many. If you go and type it in the Google, it will give you okay at least 100 journals in Nano, which is you will get.
Okay, sir. Thank you, sir. Thank you. Thank you very much, sir.
Thank you.
One madam ask some question. Please, madam, please unmute your mic and talk with our chief guest. Vengat, Abhilash, please check anybody is raising hand. This is the information for participants. There is no intention to avoid your question, whichever you send by chatting box, because of the time availability and the quality of the questions. So you are simply sending. Apart from that, we are working a lot because same question asked by several people. That time we had to combine together, and the sentence sometimes it is not good. So we have to digest by ourselves. Then we have to write it. So a lot of work we are doing offside of screen. But because of the time availability, sometimes we cannot able to read your questions. So kindly cooperate with us. In case if we are not read your questions, please feel free to send a mail to the chief guest. The chief guest will answer for you. Okay, sir. Thanks a lot for your excellent presentation, sir. And you have given very nice outline about the biomedical applications of nanoparticles from the beginning, fundamental to the advanced level. Once again, on behalf of our Department of Chemistry and myself, on behalf of our participants, it's my duty to thank you for your kind acceptance of our invitation during this pandemic period. Thanks a lot, sir. Now I hand over the session to Lakshmi.
Yeah, thank you so much.
Yes, sir.
Yes.
Thank you. Thank you, sir, for your valuable answers. With this, we have come to the end of the session. It's a time to present the vote of thanks. It's my immense pleasure to propose a vote of thanks at this memorable occasion. My heartfelt thanks and gratitude are due to the speaker for his kind acceptance of our invitation in spite of his busy schedule and his deliberate lecture. I extend my sincere thanks to the conveners, Dr. Kuttirani, Professor and Dean, School of Physical and Chemical Sciences, and Dr. Ishwaramurthy, Professor and Head, Department of Chemistry, for rendering their support in conducting the event at this pandemic period. I extend my warm thanks to our coordinator of the event, Dr. Vasimalai, Assistant Professor Selection Grade, for his efforts to organize such an event. I thank all the participants for the support and active participation. Thank you.
Okay. Thanks a lot, participants, for your kind attention. The tomorrow speaker, please announce, Lakshmi.
Feedback link has been posted in the chat box and it will be active for 30 minutes. Feedback is mandatory for certificate, so please ensure it has been duly filled and submitted. And this is to intimate that the tomorrow's session starts at 4 p.m. and the speaker is Dr. P.S. Kumar, scientist, Institute of Nanoscience and Technology, and Autonomous Research Institute of DST, Mohali, Punjab. His topic is recent trends in agriculture nanotechnology, and the participants' presence is highly appreciated. Thank you.